Hello, I'm Willie Bear ASMR, and today I'm going to be talking to you about fungi. I'm a grad student in biology, and I work in a lab that focuses on fungi. If you didn't know, the word for the professional study of fungi is mycology, and somebody who studies fungi is a mycologist. Mycology. I don't necessarily call myself a mycologist because my interests are pretty broad. I'm really interested in evolution, but fungi have some extremely interesting evolution, and I'm very happy to be in the lab I am, and I think the work we do is very interesting. But today, I really just want to give you a basic background on what fungi are, focusing on mushrooms, because most people seem to find mushrooms more interesting than other kinds of fungi. So, my goal today is to make this very, very accessible and as relaxing as possible. I know science can be kind of intimidating for some people, including me, and I really want to make everybody feel comfortable and like they can understand what I'm saying and why I'm using the words I'm using. And I don't want to make anybody feel like they're too dumb or not experienced enough to understand what I'm saying. And obviously, if you have questions in the comments, please ask, but I really will try to make this as easily understandable as possible. And if you hear some jingling in the background, that's going to be some squirrels messing around with a bird feeder that's attached to something jangly on my neighbor's house. So I apologize for that, but I don't think it's necessarily an unpleasant sound. So, as I said, mycology is the study of fungi, plural fungi, but singular fungus. So, what is a fungus? So, a lot of people have this idea that fungi are very similar to plants, and for a long time, Western science did think that fungi fell under the category of plants, but there's a very good reason that they don't. In fact, there are several very good reasons from different points of view. So, for one thing, plants photosynthesize. That means they use the energy from sunlight to make sugars that they can use for energy. And to do that, they use chlorophyll. And that's why leaves are green. I'll draw us a leaf. This nice silver gel pen. It's not a super good leaf. So you might notice fungi don't have leaves. They don't have chlorophyll either. They don't photosynthesize. They don't use sunlight to make energy. Instead, they eat like we do, more or less. They take in nutrients from things that we would consider food if we were eating them. And they do this by excreting chemicals outside of themselves, whereas we do it internally. We put the food inside of ourselves. So fungi don't fit into the category of plants based on the fact that they don't have chlorophyll, they don't live like this. But what I'm really interested in is evolution. So if we look at the evolutionary tree of organisms that are related to us, so things like fungi and plants, things that aren't bacteria, we see that fungi are actually more closely related to us than they are to plants. So if animals are right here, then fungi are right here, and plants are down here. So what is this thing? I don't expect you to know what it is. Many of you probably do. Many of you probably have some understanding, but like I said, I don't want to have to assume that you guys are coming in with any kind of prior knowledge. I just want you to be able to relax. So, this thing is called a phylogeny. That's just a fancy way of saying a tree of evolutionary relationships, or a family tree if you prefer. So, we start down here at the root, and as evolutionary time goes forward, as things evolve over time, these different lineages of organisms split off from each other. They become their own distinct groups that don't interbreed. So within plants, you'd have all these branches for different kinds of plants that split off, same within fungi and animals. But as you can see, fungi don't split off of the plant lineage. They also don't split off of the animal lineage. Fungi and animals share a common ancestor right here. This common ancestor was neither a fungus or an animal, but we share a more recent common ancestor than fungi and plants do. So this is further back in evolutionary time. So the way you read one of these phylogenies is that these two branches, you could swap them if you wanted to. So give ourselves a little bit more room. So I could draw it instead with fungi here and animals here, but plants needs to stay back here. So these two things are equally related to each other. They share this common ancestor, but that means that this whole group is equally related to plants. They share this common ancestor back here. So no animal or fungus is more closely related to plants than any other. They are all equally related to plants. They split off right here, and they've stayed separate ever since. All right, now that I've introduced the idea of phylogenies or family trees and where fungi fit in, I want to go into a little more detail about how different kinds of fungi are related to each other, and I promise we'll get to mushrooms soon. So there are lots of different kinds of fungi. You might think of mushrooms when you think of fungi. I have this nice card here and some plastic. I think that's a nice kind of sound. Kind of an old-fashioned style of illustration. Many of these mushrooms actually have really old scientific names on them that aren't the current name that you would use to refer to these particular mushrooms, but those kinds of things change over time as we learn different things about the phylogenies or evolutionary relationships of living organisms. So you might think of mushrooms when you think of fungi. You probably do. But you might also think of something like a mold. So we find molds in our fridges, on old food, and there are a lot of different kinds of molds. And in fact, a lot of fungi are things that we would consider molds. So I'm going to draw for you a very simplified fungal phylogeny with this nice, smooth silver gel pen. I love drawing with these things. They're so nice and smooth. Just glide over the paper. All right, so now we have the outline of our basic fungal phylogeny. So there are going to be some big words here, but I'll abbreviate them. And I gotta tell you, when I see a large scientific word, really long, lots of letters, I don't always know what all of those parts mean either. So I will often use shorthand to refer to that concept. So the first of these major evolutionary groups of fungi is basidiomycota. And again, that looks long. All it really means is basidio. It really just refers to this specific group. It's named for itself. And then mycota basically just means fungus. Just like mycology, myco means fungus. So all of these groups end in mycota. But I'm not going to write mycota each time because it's really long, and for these purposes, I think it's unnecessarily confusing. So the next group is called the ascomycota, but I'm just going to call it asco. In fact, that's what mycologists refer to this group as, is ascos. And this group will commonly be referred to as basidios. So I started up here in the phylogeny because these are the groups that people are going to recognize more easily. So basidiomycota contain most things that you would consider a mushroom, but also some other things that don't produce something that looks like a mushroom. Ascomycota produces a lot of molds, but also, see if I can draw anything that looks remotely. Oh, that shouldn't be a spiral. I don't know. I don't think you'll recognize this, but it's supposed to be a morel. So here's a much better morel here on this card. So ascos are a lot of molds, but also a few things, just a few, that you would recognize as a mushroom. So next down, we have a group called mycology. This contains some human pathogens, a lot of things that look like molds to us, and really importantly, some fungi that actually have a mutually beneficial, friendly relationship with plant roots. So here I've tried to draw a plant root. Then maybe here's some little fungi connected to it. They have these little tiny bulbs. So mucor has some really important plant associates, plant friends, but it also has a lot of molds and things that can actually attack humans. So a lot of diversity, a lot of interesting stuff, but nothing that we would recognize as a mushroom. And then down here, to give you an idea of what a very early diverging, so I don't want to call it primitive, but it did split off of the fungus evolutionary tree earlier than any of these other groups. I wanted to give you an idea of one of these groups of fungi. You might be familiar with chytrids. They are causing a lot of problems for amphibians right now, unfortunately. It is a fungus, but it actually has a flagellated spore. So it'll look kind of like a bacterium or sperm in a certain dispersal phase. And these are, in some ways, simpler. I really want to get those quotes in there. But in some ways, they are simpler than some of these fungi that produce these large structures that we think of as mushrooms. So in this phylogeny, as I said, we're going to be focusing on mushrooms. So I'm going to use, I have a few stickers to show you where in this phylogeny we're going to be focusing. Sticker sheet has a pretty nice noise. So, okay, I've stuck a flower right here because, as I said, phylogenies move forward in evolutionary time. This is the earliest split. These two groups, all of these guys, and then chytrids split away from each other. And then up here, there was another split where mycology, split away from these two groups together. These two groups together are the most recently diverged groups, so they evolved the most recently from the fungal lineage, and we call these Dikaryotic. So, why in the world do we call them Dikaryotic? So that's a story in and of itself. I'm going to give us a little more space on this paper. So, us humans, we have 2 copies of our genome. You probably have heard some variation of this, and again, may have various levels of understanding. But we inherit 1 copy of our genome, all of the DNA that we have in our cells. We inherit half from one parent and half from the other parent. So this is 1 genome. Say one comes from sperm cell, one parent, and the other comes from the egg cell of another parent. So in us, these two things combine. Whole genomes, all the DNA in each of these combines, and we get our genome. Integrated, all of the DNA kept together in the nucleus of our cells. Half from the sperm-giving parent and half from the egg-giving parent. However, fungi have to be special. They can't do it like we do. So, in the Dikarya, in the Ascomycota and basidiomycota, instead of combining, you get one genome from one parent, and fungi don't make sperm and egg cells. They have lots of different kinds of ways they can reproduce. You get one of your genomes from one parent and one from the other parent. Here, I'll put a dashed line here so we know these aren't the same. And that's it. They come together and they're still separate. That's a little bit of paradox there. They don't come together, but they are together in that they are the full genome of a single organism. But in the cells themselves, these two genomes remain separate. And it's okay if you don't understand why exactly that's so weird, but it is very different from what we do. And I'm going to tell you guys, nobody knows exactly why fungi do it this way. There are some ideas, but it is very weird. They are one of the only organisms on Earth to do this. So we know that these two groups, Ascomycota and basidiomycota, are united by having this dikaryon. So the dikaryon refers to keeping these two genomes physically separate in the cell instead of combining them. So if you want to picture how these two separate genomes, physically separated from each other, look in a fungal cell, I'll make you a little diagram. So again, this only happens for Ascomycota and basidiomycota, which produce things that look like mushrooms. So this is a really long, really thin piece of fungal material called a hypha, plural hyphae. Here, now that I've made it plural, I'm going to have to draw another one because it wouldn't be right to only have one hypha when I've written hyphae. So this is called a septate hypha because it has these walls. They're called septa. They're also called walls. That's a fine thing to call them. So each of these little compartments is its own cell. And each of them has one version of each genome, each copy of all of the DNA. So that's what a dikaryotic hypha looks like, more or less. When it grows, it extends out from this tip. And there's a fancy mechanism that forms in some basidiomycota and some fungi that are mushroom forming or closely related to mushroom forming fungi. They form these fancy structures called clamp connections. You can see those under a microscope. Really, just what the clamp connections are for is as this tip extends forward, it helps make sure that one copy of each genome, each nucleus of DNA, gets into each new cell as it puts a wall behind the extending tip. So I could diagram out for you all the steps of the process, but I think the important thing to note is that each of these need to be in each one of the cells, and they kind of just travel through this little tube to make sure that one copy is in each cell as they form. And then after they're done, the tube's just kind of there. It doesn't seem to do anything. This is another kind of mysterious yet basic thing about fungi. Lots of things like that. But that's called a clamp connection. And then this hypha, if it doesn't have walls, it's called cenocytic, which again is another one of those words that you don't need to know. It just means no walls, no septa. And lots of fungi that aren't ascomycota or basidiomycota, the guys that have two different separate genomes and produce mushrooms. Most things that look like this are not in those two groups. They are simpler or more primitive fungi. Although, again, I think those words are confusing because they don't always refer to really the evolutionary reality of the situation. They're a little bit of an assumption. But this is what the structure of fungal tissue looks like. All these little thread-like interwoven hyphae. And when you have a lot of hyphae grown together, these things are microscopic. They're branching off each other. They're going back into each other, which is called anastomosis. Literally just means going back into each other. Again, not important for you to know. But a lot of fungal hyphae together probably won't look much like this. I don't think I've drawn it very well. But all together, lots of hyphae from the same fungus are called a mycelium. And that's the singular. So mycelium is what all fungi that aren't yeast look like as they're growing. Maybe they'll produce a mushroom. Maybe they're a kind of fungus that doesn't produce a mushroom. But they'll all look something like this. The difference between this guy and a yeast is that instead of growing in thin, stretched out hyphae, a yeast buds apart into separate cells. Here we can say we start out with one yeast cell right here, and then it buds apart or splits apart in some other way, and then we get these two separate yeast cells, and they just live together in just a big clump. So that's what a yeast is. Also fungus, it just doesn't grow like this. All right. New piece of paper. Okay, so now we know some of the basics of fungal evolution and how fungi grow and what fungal tissue is made of. And now we can start talking about mushrooms a little bit. So, like I said, mycelium is a bunch of interconnected hyphae. In this case, let's say they're growing underneath the ground in the soil. Probably decaying organic matter, like parts of plants, dead animals, other fungi. And like I said earlier, they're doing this by actually exuding, producing these chemicals that help them break down food into separate nutrients that are then absorbed into the mycelium, whereas our digestive chemicals are housed in our tummies, and we don't need them to go anywhere. We just bring the food to them. So here's a not very good mycelium. It's going to be way threadier and more complex than this. But once a mycelium reaches a certain mass, probably different under lots of different conditions and for different fungi, it might produce something called a mushroom. Oh, that's kind of off-center mushroom, but you know what? It's nature. Most mushrooms aren't perfect. They're still beautiful. Oh, that's pretty lopsided though. All right, let's call that good. So this mycelium has produced a mushroom. What do we know about this mycelium by the fact that it's produced a mushroom? So, for one thing, we know for sure that it's a dikaryon. So, I said the word dikarya meant that it has two separate nuclei of DNA, two separate genomes that don't fuse together. So this only happens when two different fungi have already mated with each other. So this is going to be a little bit confusing. So we started out with one mycelium. It had only one genome. It grew from a spore. Here's my spore. Sometimes they've got cute little spots. It grew into mycelium. It only has one genome. But then we get another mycelium. Sorry, I'm getting lazier and lazier with this. And it's only got one genome too. It grew from its own spore. So how do you get a dikaryon that has two nuclei, two different nuclei? So these two actually do come together and mate. They merge with each other if they're compatible, which is its own story that I don't think I have time to get into here. But some fungi have lots and lots of different sexes, essentially. And if they're compatible sexes and they can mate with each other, they form a dikaryon with both kinds of genome, both kinds of nuclei in every one of their cells. So once you're dikaryon, then you get to make a mushroom if you want. I guess it's up to you as a fungus. The other thing that we know about this thing is that it's a basidio. This is a term from that fungal family tree that I just showed you. Oh, I just totally misspelled that. Talking and writing. Basidio. Mycota. Let's just cover that up so we don't get confused. All right. And I'm going to be referring to these fungi just as basidios. So only basidios have a mushroom that looks like this with these gill structures. The other option is ascos, like I mentioned, but they can't form something that looks like this. So let's start talking about the difference between basidiomycota and ascomycota. So both of them need to form a dikaryon in order to reproduce. But basidiomycota can look all kinds of different ways. We can have, like I drew before, these gilled mushrooms. We could have truffles. The basidio truffles are not the really nice, yummy truffles that you might be thinking of. But they do form truffles. They're kind of ugly looking. There are also basidio mushrooms that look like little corals. I work on some of those. And there are basidios that don't form mushrooms at all, but there's a huge variety. But then in ascos, you're really only going to find a few types of things that look like a mushroom to you. One, as I mentioned earlier, is morels, and I'm determined to draw a better morel this time. Yeah, it is what it is. The really yummy, expensive truffles that people train dogs to hunt for, those are actually ascos. So they're not very closely related to these guys at all. And then there are lots of cup-shaped fungi in the ascomycota. There are some of these growing in the tropics. You might find something that looks like this growing in your shower or in your gross basement. There are a lot of different fungi that look like this. So what's crazy is that you might think that something like a morel seems like it has a cap and a stalk is more closely related to something like a gilled mushroom, but it's actually more closely related to all of these. Because remember, when basidiomycota and ascomycota, basidios and ascos split away from each other, that meant that they were distinct groups forever. They don't interbreed whatsoever. So all of these had to come out of ascomycota. And all of these had to come out of basidiomycota after these two groups had split. Even these truffles, they're not very closely related to each other at all. This truffle is more closely related to all of these friends than it is to any of these, even that truffle, even though they look the same. So fungi are really good at evolving the same forms over and over again. That makes it really confusing to figure out their evolutionary relationships, because you might think that these two things were more closely related, or these two. But we know that they aren't, because we use DNA evidence, which is more reliable. by far. And I can discuss that in a different video, but that's a big subject in and of itself, like so many things about fungi and biology in general. All right, now I promise we're going to focus in on mushrooms. So we can move this up. I'm going to show you a relative of mushrooms. It's within Basidiomycota. It's Basidio, but it is not a mushroom. So I have this book. It's called Pests and Diseases, and I got it from one of those little free library boxes. We have some in my neighborhood, and I figured it'd be a nice thing for me to own. It's kind of vintagey. It's got some cool pictures of bugs and fungi. So I've got a page bookmarked here. All right, so here we've got a leaf. It's been infected by a fungus. This fungus is actually a basidio. It's closely related to the kinds of basidio fungi that produce mushrooms, but it is not part of a group of mushroom-forming fungi. I'm trying to keep from having to just show you guys a million phylogenies, a million family trees. So I'll just say that this thing branched off before mushrooms evolved, before mushrooms evolved, before basidio fungi evolved to begin producing mushrooms. It's called a rust. It's in the group Mycotina. It's a very long name. You don't need to remember it. But this is a rust. You've probably seen these kinds of spots on leaves many, many, many times. And very often, they're actually rust. It's pretty closely related to mushrooms, because it is a basidio. Ironically, I did bookmark it with this 10,000 colones note from Costa Rica, and there is a fungus here. This little cup guy, I was very excited when I noticed this. It's a coquina, and it's actually an asco. So remember, we have these two groups, Basidiomycota, basidios, and Ascomycota, ascos. They can both form things that look like mushrooms, but they're not very closely related to each other. So this thing is an asco, so it's more closely related to morels and truffles than it is to any other thing we think of as a mushroom, because it's an asco, not a basidio. So we also have another representative. Oh, that's some jangling. It's a squirrel running around out there trying to get some bird seed. We have something else that is a basidio, but it's not mushroom forming. It's not part of a group that can form mushrooms. And this is called a smut. This one is probably the most famous smut, corn smut, Ustalagomatus. It's edible, and it's considered a delicacy in Mexico. I've never had it. I really badly want it. I think it looks delicious, but I kind of like eating weird things. So I know it might not seem like everybody's cup of tea, but I bet it's really, really good. So this thing infects plants like rusts often do. A lot of smuts grow in grasses, and corn is actually a grass. These are part of a group called Ustalaginomycotina, which is also unreasonably long, and you don't need to remember it. But this thing is a basidio. It's related to mushrooms, but it can't produce a mushroom. All right. So we've heard a lot about stuff that can't produce mushrooms. So what can produce a mushroom? So I drew you a little rudimentary diagram of a lot of these different possible forms for mushrooms. Up here, we have gills under the cap. We have a round thing like a truffle, and we have some wacky things like corals. So I've got this nice book, The Kingdom of Fungi by Jens Petersen. All these like coffee table books, I think, have a really nice tapping sound. So you can even see on the cover of this that we've got some gilled mushrooms. I think this is the genus Marasmius. They're very small, very pretty. So this is a very common kind of mushroom you'll see, this gilled type. Not all gilled mushrooms are very closely related to each other. A lot of mushrooms have gills. So the spores are actually produced on the sides of the gills. So I think I've bookmarked some more examples of gilled mushrooms. Oh yeah. Very, very beautiful. Lots of different kinds of gills. This is a trait that was used to identify mushrooms for a long time, is the way the different gills looked. It's still used. Obviously, we don't really use it for making phylogenies in science. We use DNA, as I said before, but when you're out in the field identifying fungi, sometimes the gills are important to identifying whatever you've found. So here are some really nice different gills, lots of different colors. So the mycological, the fancy scientific term for gills is lamellae. Lamellae. So that is one type of structure that a mushroom can have under its cap. So here is a silly little Mario mushroom, little life mushroom that I have. It used to have mints in it. I was actually given to me by a student in the mycology class I TA'd for my boss sometime last year. Yeah, time is meaningless. But it's cute, and I think it has a nice sound. And the cap comes off, and then we have one mint left in there. So I have this here because I think it makes a nice noise, and also because I just wanted to establish the idea that in mushrooms, spores, the way that mushrooms reproduce are always coming from under the cap. They might have different structures under their cap, like lamellae, but that is where all the spores are produced in something that is a cap and stalk mushroom. Cap, stalk. And those have fancy names too, but I don't see any reason to use them. So something else that mushrooms can have under their cap besides gills is pores. So this is a mushroom I made out of air-dried clay, and it's supposed to be a porcini. It's a very popular edible mushroom, but you can't farm it because it can't live without, well, it can't fruit, it can't produce mushrooms. It can exist, remember, as our mycelium, but it can't produce mushrooms unless it's in a special relationship with the tree. And we haven't figured out how to do that in a farm or lab. So you only get these things wild harvested, basically, but they're delicious. And so I was trying to make models of them for fun. And instead of gills under the cap, we have pores. So on a real mushroom, each of these pores goes back in to the underside of the cap and produces spores that then fall out of the pores. They're spores and pores. And then go off to grow and become their own mycelia. Plural mycelia, singular mycelium. So this porcini is definitely a mushroom. It's a basidio, but instead of gills, it has pores. Kind of funky looking, but there are a good amount of other mushrooms that have those pore structures instead. So I tried, when I was making this guy, I tried to get the stalk right. They have this really thin, fine sort of netting structure over their stipe. It's called reticulation. I absolutely couldn't get it right. I'm not super good with clay. This is one of my first real projects with clay as an adult. But I made another porcini for my boss because he's a big porcini guy. Worldwide porcini expert is probably more accurate. But I made another for him. This was Mark II. The one for him was Mark III, and it did have better reticulation. But this one, you can't really get the idea of how to tell a porcini apart from a close relative from this, but I think it looks a good amount like a porcini. Walking along in the forest and you see a cap like this, it's kind of like a hamburger bun on the ground, and you know you've just found a delicious porcini. All right, so I think I've given you a basic idea of some of the different forms mushrooms can take and what they're related to. So now I want to zoom in a little bit on something very, very important to mushrooms and to fungi in general, is spores. So what is a spore? Spore is just a way of getting your genetic information around. So in basidios, you're producing a sexual spore that has one genome in it. And that, of course, will find another, they'll grow into mycelium and find a spore that's grown into a mycelium, and those will come together, and then they'll have two genomes and be a dikaryon. But for now, we're just a spore. We've just got one genome. So spores are produced in basidios. So from now on, I'm only going to talk about basidios. Just don't want you to be confused. These are fungi that are majorly mushroom forming. Many of them form mushrooms, but not all of them. So basidiospores are produced by a very special structure. Drawing a very rudimentary version of it because I don't want to get messed up. So remember when I said earlier you might not remember, and that's okay. I talk a lot. I mentioned that basidiomycete are kind of named for themselves. And that's because they're named after this basidium structure that produces basidiospores. I know this looks really arcane and mysterious right now, but it will make sense. So these are basidiospores. So this whole deal is super microscopic. So you would have, let's say in a guild mushroom, this is the underside of the cap. This is where the stalk connects, and these are the gills. So we're going to take one of these gills and we're going to turn it on its side, do a little 3D art here. So if you can picture that, one of these gills, this long sort of, I don't know, like it's flat. I'm picturing it a little bit like a sheet of paper here. And this is a large magnification. And then these basidia, that's the plural, the singular is basidium, are growing off the side, the broad face of these. So in here, if you could look under a mushroom and actually see the basidium, it would actually, I don't think you can see my drawing here, but they would be all on the sides of these things, of the gills, or as I mentioned earlier, lamellae. And there are other types of ends of hyphae, those little tiny fungal threads that grow on the sides of these gills, but basidia are very important, and we're going to be focusing on them. So this thing could be one of these little tiny things. So this thing is the basidium. Its job is holding spores. And since it's a basidium, the kinds of spores it holds are basidiospores. Most basidia are going to hold 4 basidiospores, but not all. Some of them have 2 for some reason. So some basidios have a basidium that looks like this with 2 basidiospores. All right, so these are our little spore friends, some little dots. Okay, so we know that these spores are on these weird crown structures on the side of a gill. But how do they get out from the underside of the cap to be dispersed and go grow into their own mycelium, and then find another mycelium and combine and maybe someday make a mushroom? So this is one spore, again, one genome, half of what it will have once it's mated. And it's got this like kind of little layer of water around it, and it has a drop of water right here. And this drop is called Buller's drop. Now, this is a really cool fun fact about fungi. I'm going to give this guy just some spots, because often, again, spores will have spots or other ornamentations. They're not always smooth like this. They can be prickly, rounder, more irregular, less irregular, and there's very little known about why different spores look different ways. This is a character that actually has been like somewhat useful in seeing which fungi are more closely related to each other, which mushrooms are more closely related to each other. But again, ultimately not as reliable as DNA, but spores have a lot of variety and can look lots and lots of different ways. And sometimes when you're trying to ID a mushroom, looking at the spore can be really helpful, but you need a good microscope. So this Buller's drop is water. I'll color all this. Well, maybe that'll be confusing because I've already given it spots with silver. But all this is water. And then at some point, due to humidity conditions or something else, this drop will jump onto the water coating the rest of the spore. And this process isn't perfectly understood, but it was actually discovered by this guy Buller like over 100 years ago. So pretty impressive work by this guy. So this snaps onto the rest of the spore, and that force actually generates a ton of force. Like I think people are always comparing it to like it's G's of force, like a rocket taking off. So it generates all this force, it snaps, and it propels the spore away from the mushroom. So the way I always used to imagine this before I started carefully studying mycology is that the spore, so remember, we're looking under the cap. So the spore, the Buller's drop snaps, and the spore goes like it just shoots away from the mushroom, because why else would you have that much force? Why would you need that? But this is not true. That is not true. That's not what happens. Instead, we're actually looking, so, all right, so bear with me again. So we have under this mushroom cap, we have these gills, and between the gills on the gill faces is where these spores are produced in those basidia, those weird crown-like structures. So we're going to take some of these gills and now pretend that we're looking at each of these gills head on from its thin side. So these are basidia being produced on the flat face of the gill. All right, so, and let's say this is oriented so that this direction is down. So a basidiospore gets ejected because its Buller's drop snaps and generates all this force, but actually all that does is it shoots it out, just boop, just right into the space between these two gills. And then it just kind of falls out. It's so light and tiny it gets carried on the wind. So all that force just to shoot it out in the space between these two gills. And that seems like it's worth it for the mushroom, evolutionarily, because it makes sure that the spore doesn't get stuck to all these basidia and other spores. it actually makes it out into this empty space. I mean, and the spore is tiny. This space would actually be, I mean, it would be very wide if the spore were like a little dot like that. But it falls out into the space between, and that's what's important, is that it shoots out into the space and then it falls out from between the gills, and it can go and find its own place to grow if it happens to land somewhere where the conditions are good enough. So I think this is a really cool story, but I think it's hard to picture because you picture this mushroom just pew pew pew, just shooting these things out. I think this is all I'm going to tell you guys about today. I might do another video. I'd also love to do videos that are more focused in, but this is my first time trying something like this, and so I feel like this is a good trial run. I'll give you guys a little bit more tapping to end it out. This guy is Sculpey air dry clay around a foam, just like a styrofoam base that I carved. I just painted it with acrylics, and the cap is nice and shiny because I put Mod Podge on it. Mod Podge is just a really nice sealant. I use it for everything. I'm not sure if you're supposed to use it for everything, but I do. All right, thank you so much for listening. I hope this was relaxing and easy to follow. I'd love tips so I can improve in the future. And I look forward to seeing more of you guys soon.